Yes, Shalini, very good evening. Am I audible to you all? Yes. Good evening, Subhan. Yes. Yes, Sanjay, very good evening. Is the slide is visible to you? Yes. Very good evening to all of you. Happy to see you all. Yes. Shall we start the class today? Meiosis. I hope you all remember mitosis. And today we are going to discuss meiosis. Yes, sir? Shall we start with a question, my dear children? Meiosis. Are you all ready? Are you all boosted? Yes. Very happy evening to one and all. Shall I start the question with a small question? Yes, Sanjay. See, a plant has 10 chromosomes. Okay. A plant has 10 chromosomes means in next G2 cycle, okay, what is the number of chromosomes we may have? See, next G2 cycle, number of chromosomes we may have is how many? See, total number I said it is 10. Okay. And what would be the number of chromosomes we may have? So, 10 chromosome I said. Yes, are you all thinking? I said number of chromosome is 10. What is the number of chromosome in next G2 cycle is the question. What is the number of chromosome in next G2 cycle? Yes, Arishakti, very correct answer. Excellent answer, Arishakti. It is 10. Okay, because in G2, of course, chromosome is not going to duplicate whether it is mitosis or meiosis. If I said DNA, it might have doubled. Yes or not? Yes, Tiruvila, excellent answering. Yes, it is. It is 10. Very correct answer, 100% correct answer. Thank you for all joining and attending the answer. Yes. Fine. Let me start the meiosis. Okay. What's the other name of meiosis, children? What is the other name of meiosis? What is the other name of meiosis? Yes. Yes, Tiruvira, excellent. And yes, Subhan, excellent, Subhan. Very correct answer, 100% correct answer. It is reduction division. Yes, Hari. Shakti, it is reduction division. Very correct answer, Hari Shakti. Excellent, ma'am. Okay. So, meiosis means reduction division, as you all know. And meiosis, we have meiosis 1 and meiosis 2. Okay. So, we have meiosis 1 and meiosis 2 is there. See, my pen tab is so urgent. It's writing very fast. Meiosis, I hope you can understand. Meiosis 1 and meiosis 2. Okay. So, we have meiosis 1 and meiosis 2. And this meiosis 1 is otherwise called reduction division. Meiosis 2 is called equatorial division. Okay. Meiosis, the term has been coined by which scientist? Any idea? Who coined the term meiosis? Who coined the term meiosis is Farmer and Moore. So it is Farmer and Moore actually. So let me write like this anyway. It is Farmer who coined the term. 1 second, children. I hope you can all see my slide now. So it is Farmer who coined the term actually meiosis. Can you all tell me? Yeah, Farmer and Moore is very correct answer. Tiruvira again. Can you all tell me like meiosis happens where and all? Where and all meiosis is possible? Yes, Shubhan. Yes, Sanjay. Yes, Shalini. Anybody? Meiosis is possible where and all. We have so many tricky questions from meiosis. Yes, of course. Can anybody answer me? Where and all meiosis is possible? It is possible, of course, in gametes. And it is possible, of course, yeah, in reproducing organism is correct answer. Of course, in gametes, yes. Gametic meiosis, we are forming sperm and ova because of meiosis only. Yes, sir? Sperm and ova. Of course, sporotic meiosis. We might have discussed sporotic meiosis in bryophytes and pteridophyte children. So in bryophytes and pteridophytes, sporotic meiosis is possible. I hope you remember the spore undergo meiosis and then it will be degazed. Then we have zygotic meiosis. The zygotic meiosis is possible in algae, chlamydomonas. Yes, sir. So meiosis is even possible apart from gametes in spores as well as in zygote. Fine. So meiosis, we have two meiosis we discussed, 1 and 2. Meiosis 1, we have prophase 1, metaphase 1, anaphase 1 and telophase 1. Can you tell me children which is the longest phase? Is it prophase 1 or metaphase 1 or anaphase 1 or telophase 1? Which is the longest phase? The longest phase of meiosis is prophase 1 or prophase 2 or metaphase 1 or anaphase 1 or telophase 1. Yes, Dharini, very good evening. Happy to see your name. Yes, Tiruvira, very correct answer. It is prophase 1. Is the correct answer. Very correct answer. It is prophase 1. Okay. Fine. Now let me ask you one joke. Let me see who is answering. Okay. Now, a cell wants to convey information to another cell. How it will do? A cell wants to convey the information to another cell. How it will do? A cell wants to convey the information to another cell. Yes, of course, very correct answer, Dharini. Excellent. You're proving you're my student, okay? Dharini, yes, it is via cell phone. Yes, Prakash, that was the correct answer. Praveen Kumar, correct answer. Prophase 1, yes, cell phone is a very correct answer. Yes, cell communicate to the next cell through their cell phone. Yes, fine. Very correct. Fine. You know now, let us proceed to the class. Prophase 1 is the longest phase, as you all said. And we have, before prophase, interphase is there. So, interphase is happening here. Yes, Sivaprakash, that was the correct answer. Cell phone is the correct answer. Yes. Interphase. Okay. Is happening. Yes, you are my student answering all these jokes also. Very correct. Yes, interphase is happening here before prophase. So we will be having G1 phase where we synthesize ATP as well as RNA, and also S phase where we double the DNA, G2 phase where we synthesize tubulin protein, and then of course we are undergoing meiosis now. So this G1, yes, and G2 we discussed in the last class itself. So we are going to discuss now about meiosis. So we are proceeding to the prophase. Prophase 1, can you all tell me what are the stages involved in prophase 1? How many stages are there? Is it 3, 4 or 5? Or 2 or 1? Yes? How many stages are there in prophase 1? Yes, Tiruvira, very correct answer. It is 5. Very correct answer. Excellent, Tiruvira. So we have 5 is the correct answer. Yes, Tarunika, you are very happy. Yes, Subhan, very correct answer. 5 stages. As you know, it is leptotin, zygotene, pachytene, diplotene, diakinesis. So this is what very important, children. Questions will come like match the stages, etc. Now, can you tell me where do we find thin uncoiled chromosome? In which stage? Thin uncoiled chromosome we find in which stage? Among the 5. Thin uncoiled chromosome. Yes, my dear. Siva Prakash, Sanjay, Praveen. The uncoiled thin chromosome we find in which stage? Is it in leptotin or zygotene or pachytene or diplotene or diakinesis? Yes, Pa? Any guess? Yes, Tiruvira, again very correct answer. It is leptotin. Yes, it is leptotin. 100% correct answer. So you can see in the picture also, they are thin, long, uncoiled chromosome. And if you see, they will be attached to one end generally, and they are free on the other end. Okay. So when they are attached to one end, free on the other end, it looks like a bouquet. So the stage, even we call it as bouquet stage. Okay. So leptotin, where we find thin uncoiled chromosome, and it will appear like a bouquet. So we call it as bouquet stage. Yes, it is leptotin. Subhan, no doubt. It is leptotin only. So possible question, thin uncoiled chromosome we found in Which stage of the meiosis? Okay, it is in leptotene. Now next one, zygotene. What are the possible questions? We find pairing of homologous chromosome. So pairing of homologous chromosome is happening here. Okay. This pair we call it as what actually? We call it as bivalent chromosome. Yes or not? So bivalent chromosome. And you know very well, inside that bivalent chromosome, assume that this is a bivalent chromosome, you can find that double the number of chromatids. So chromatids will be doubled. I am drawing the dotted lines to make you understand. So usually they'll make it like this, or generally in books they'll draw like this. In bivalent chromosome to indicate chromatid. Okay. So these are chromatids. So when you are having double the number of chromatid, it is either called dyad stage or tetrad stage. So you should know the dyad and tetrad condition. Okay, are you clear about this? I hope. For example, in human, we have 46 number of chromosome means how many chromatids we have in zygotene? How many number of chromatids we have in our zygotene? The number of chromosome is 46. 46 chromosome is there means number of chromatids in zygotene. Yes, children. I am going very slowly because this is what critical part. Anybody, any guess? Yes. Any guess? Subhan, Tiruvira, Tarunika, Sivaprakash. It is 46 or 92. 92 is the correct answer, Tiruvira. Yes, Subhan. Excellent, Subhan. It is excellent answer, 92 chromatid. So 46 chromosome means 92 chromatid is the correct answer. Because you know DNA will get double in S phase, not the chromosome. So you will have 92 chromatids, 46 chromosome in the bivalent chromosome, of course in zygotene stage, to indicate that 92 only the doubling. We are showing like a dotted line, or you can show like this structure also. Both are correct. I hope you understood zygotene. And when pairing happens, the condition we call it as synapse. Synapse or synapse. You can pronounce anyway synapse. So they are paired. Can you see the blue ones are paired here and the red ones are paired here? So pairing of homologous chromosome. How this pairing can be done? The glue is synaptonemal complex. So synaptonemal complex only helps to glue the homologous chromosome. I hope you all understood how we got the 92 chromatid. Any doubt? Shall I proceed? Now next stage we are proceeding to pachytene. So in pachytene we have the important step called crossing over happens. So crossing over means what? Exchange of genetic material. Yes, ma'am. Exchange of genetic material is happening here, or shuffling of genes will happen. This crossing over is very important. You know, one enzyme is participating in crossing over. Can you tell me, children, what is that enzyme that participate in crossing over? The enzyme that participate in crossing over is what? Yes, Shiva Prakash, Tiruvira, Shubhan, Tarunika, Dharni. So, what is the enzyme that participate in crossing over? Yes, Tarini, very correct. Excellent answer. Yes, Tiruvira. It is recombinase. 100% correct answer. Okay, so the enzyme is recombinase enzyme that participate in crossing over. Okay, so exchange happens. Of course, shuffling of genes happens between non-sister chromatids of homologous chromosome. So, non-sister chromatids of homologous chromosome. Can you see that I have shown you here one blue color pair and red color pair. So, the blue is exchanging with the red. They are exchanging their gene. Shuffling is happening with the help of recombinase enzyme. Okay. And this is what very important for variation actually. So only though the parents are same, the brothers or sisters or brother and sisters of the same family are not identical. Shuffling happens. Okay, I hope you all understood. So even let us learn like this. Let us take, we have taken color balls in a bowl. Maybe red, blue, green, yellow, orange, something. So the shuffling happens. We have hundreds of colors. So some genes picked by us and some genes picked by the others from the parents. So only this variation happens, and variation is the raw material for what? Variation is raw material for what? Yes, Dharini's friend, very good evening. Welcome you, hearty welcome to our class. Yes. So, what is that raw material for variation forms? Raw material for what? Yeah, very good, Dharini. Excellent answer again. Evolution. Yes, Bharta Sardi. Very good evening. Evolution is the correct answer. 100% correct answer. Evolution. Okay, so these are the possible question. Of course, crossing over happens in which stage? They may ask you. And of course, the enzyme which participate in crossing over, another possible question. And of course, variation is raw material for evolution, as you know. And exchange happens between non-sister chromatids of homologous chromosome, maybe a statement type question. Okay, I hope you are all clear with the pachytene. And next stage we are proceeding is diplotene. So diplotene, if you see the place where the exchange happen alone is retaining. The stage we call it as what? So the X-shaped place where the exchange happens retain is called dash. Is called anybody? The place where the exchange happen alone, you can see it is retained. Other places, they are all separated because of the dissolution of synaptonemal complex. Yes, it is chiasmata, Dharani. Very correct again, chiasmata. The exchange structure where you find is called chiasmata. Is the very correct answer. Okay. And dissolution, of course, you know, it is dissolution of synaptonemal complex. So synaptonemal complex dissolve everywhere except the place where the chiasmata is present. So chiasmata retaining is the important stages in diplotene. Okay. Then next one, we are proceeding to diakinesis. So in diakinesis, the important step is terminalization. Terminalization is unzipping of chromatid. Yes, pa. Unzipping of chromatid. So we are removing the nicking. Okay, like the broken pieces, the exchange material are going back to their original places is what we call it as terminalization. So we are removing the cross, even the chiasmata point, unzipping or dissolution happens. This is what we call it as terminalization. Yes, Ahmed. Very good evening. Happy to see your name, Ahmed. Welcome you for the class. Ahmed. Okay. So we are discussing actually meiosis 1 where we are in diakinesis part, Ahmed. Okay. So terminalization is happening here. So the chromatids are getting separated. Dissolution happens fully A to Z. Like you remember unlocking your zip of the bag, school bag. So when you unlock your zip, it becomes two separate stand or not. So the same thing is happening here also in diakinesis. And all prophase events are carried out here. So nuclear membrane disappears. Thank you, thank you. I didn't understand your name. You can even give your name, my dear child. Thank you for your appreciation. It helps me a lot. It means me a lot. Thank you so much. Yes, Mr. Ajay, Pushpa Kumar. Yeah. Very welcome you for the class. So we are discussing meiosis 1. Thanks for your presence. Okay. Nuclear membrane disappears and cytoplasmic organelles also disappears. Of course, the centriole migrate to the opposite pole and spindle fiber formation happens. So these are the events that are happening in diakinesis. It is like the prophase events are carried out only in diakinesis. I hope you are all clear with this. Understood about prophase 1. Then metaphase. Let me ask you a question. See, I am giving you an onion root tip. Okay. And a shoot tip. And the flower bud. Among the three, which you will select for studying meiosis? Either it is shoot tip or root tip or flower bud you will select for studying meiosis. Yes, anybody? Can you answer me, children? Yes, Ahmed. Which one you will choose for studying the meiosis? It is onion root tip or shoot tip or flower bud? Yes, Subhan, very correct. It is flower bud. Flower bud is the very correct answer because meiosis is reproduction. Okay, I asked this. Very sorry, Dharni. Anyway, I forgot. Okay, let me ask you one another question. Fine. See, if I wanted to study the number of chromosome, I ask you to find the number of chromosome. Find the number of chromosome. Which stage you will select? Yes, Agamat, very correct. It is flower bud you will select. If I wanted to count the number of chromosome, okay, which stage you will select? Yes, Subhan, it is meiosis flower bud. Very correct answer. Excellent answering, Subhan. If I want to ask you to count the number of chromosome, which stage you will select? Either it is prophase, or it is metaphase, or it is anaphase, or it is telophase. Okay. Good evening, Pranav. I am very fine. I know you are Pushpa Kumar's friend. I remember still where you are absent for last 2 weeks, Pranav. I missed your Josh, Pranav. Okay. For mitosis, onion, root tip, and meiosis, it is flower bud. Is the correct answer, Dharini. Actually, metaphase is the correct answer. Agamad, you're rocking. Yes, it is metaphase. We will count the number, structure of chromosome, everything. Shape alone, we will do it in anaphase. Either it is metacentric or submetacentric or telocentric or acrocentric. All that you can learn only in the anaphase. The number of chromosome or banding pattern, etc. can be studied in metaphase of the cell division. Yes, children, I hope you are clear now. So, metaphase is used for studying the chromosome number and structure. As always, you can see the chromosomes are aligned in the equator and they are attached. Spindle fibers are attached to the centromere. By kinetochore. At kinetochore they are attached. Kinetochore is a disc-like structure. You can find it here. It is attached to the spindle fiber. And they all aligned in the line, in the middle line. It is called equatorial plate. Yes, Pranav, I remember you. Oh, you have some work. Fine, fine. Very good. Okay. And they are arranged in equatorial plate. Okay. Now for the sake of Pranav, one question. Let me see whether Pranav answers me. Okay. Yes, everybody can try. See, one cell stamped on other cell. What the other cell will do now? It is shouting. Can you understand my question? One cell stamped on the feet of another cell. Okay. Yes. Come on, Pranav. Answer me. See, one cell stand on like stamped. Okay. Yes. Today it is Agamad day. He is answering. Yes, it is mitosis. Yes, mitosis. We can say no mitosis is the correct answer. Yes, Subhan. You are thinking, yeah? See, Agamad rocked it today. It was the day of Agamad today. Yes, it is mitosis is the correct answer actually. So it was shouting that mitose. What? The question is for Pranav only. What Pranav? Yes. Very correct. Yes, Dharani. You missed it. Okay. So metaphase 1 we studied possible question. Of course, they are in equatorial plane and you can study either the number or the structure of chromosome. Yes, while Pushpa in the class. Yes. Pushpa means not flower. Okay. It is fire. Yes, sir. Oh, it reduced Pushpa on it. So he is so respectful, Pushpa, to you. Okay. Sorry. Fine. So equatorial plate, they are aligned. Okay, fine. Anyway. Oh, you are really feeling sad. So you should allow him not to answer and all. Why you are threatening him? He is waiting for your response. What this Pushpa? Yes. You could have allowed him, na? This is very bad on your part. Yes. So now anaphase 1. Anaphase 1 ல என்ன ஆகும் அப்படின்னா as you know the spindle fibers short ஆகும் and do we split the centromere? Splitting of centromere நடக்குமா? Splitting of centromere, does it happen or not? Who will answer me? Splitting of centromere, will it happen or not? Yes? No, very correct. Splitting of centromere does not happen. Yes. Splitting of centromere does not happen is very correct. Yes, Pushpa Kumar, very correct answer. Excellent. 100%. அப்போ உங்களுக்கு குரோமோசோம் டபுள் ஆகாது. ஓகேயா? அப்போ நமக்கு ஆல்ரெடி எவ்வளவு குரோமோசோம் நம்ம படிச்சோம்? 46 குரோமோசோம் படிச்சோம். 92 குரோமாட்டிட்ஸ் படிச்சோம். இல்லையா? இப்போ நமக்கு ஆப்போசிட் போல்ஸ்ல மூவ் ஆகும்போது எஸ் இட் ஹேப்பன்ஸ் இன் anaphase 2 இஸ் வெரி கரெக்ட் ஆன்சர் அகமது. இப்போ anaphase 1ல நமக்கு என்ன ஆகும்? இங்க 23 குரோமோசோம் போய்டும், இங்க 23 குரோமோசோம் போய்டும். குரோமாட்டிட்ஸ் எவ்வளவு இருக்கும்? 46 chromatid on upper pole, 46 chromatid on the lower pole. I hope you are all clear. You all understood? Yes, my dear children. Parthasarthi, Dharani, Tiruvira. All got it? Shall I proceed? So, இங்கே உங்களுக்கு splitting of centromere நடக்காது. அப்போ chromosome double ஆகாது. அப்போ இருக்கிற chromatids பாதி பாதியா மேல போறது கீழே வருது. chromosome உங்களுக்கு அதே நம்பர் தான், ஏன்னா chromosome நம்ம டபுளே பண்ணல. சரியா? So, 23, 23, 46, 46 tallied ஆகுது. Anaphaseல இதுதான் questionஆ possibleஆ கிடைக்கும். Chromosomes and chromatid questions numbers வச்சு கேட்கறது. And you know very well, இங்க நம்ம வந்து அந்த shapes of chromosome, metacentrica, acrocentrica, telocentrica, அந்த மாதிரியான types எல்லாம் we can able to understand and study using anaphase 1. ஓகேயா? மற்றபடி இது anaphase லேட் anaphaseல ஒரு furrow ஆல்ரெடி நம்ம டிஸ்கஸ் பண்ண மாதிரி ஒரு furrow வந்துரும் in case of animal cell. Okay, and telophase they become two daughter cell each haploid in condition. So we are getting haploid cell. Okay, and two daughter cell they are. So two daughter cell form ஆகுறாங்க இங்க. சரியா? I hope you are clear. They are haploid in condition. Of course, in the condition same. நம்ம already படிச்சிருக்கோம். Cytokinesis proceeds with the karyokinesis. In case of animal cell, in case of plant cell நீங்க பாத்தீங்கன்னா ஒரு செல் பிளேட் ஃபார்ம் ஆகும். அந்த செல் பிளேட் அப்படி கிராஜுவலா எக்ஸ்டெண்ட் ஆகி ரெண்டு செல்லா மாறும் அப்படின்றது in case of plant cell அதே கண்டிஷன் இங்கேயும் நமக்கு அப்ளை ஆகுது. So we have already got two haploid cells. அடுத்தது prophase 2. Now tell me children, do we have interphase between prophase, that is telophase 1 and prophase 2? Do we have interphase between yes, anybody? No is the correct answer. So there won't be any interphase. So what will be existing? There will be interkinesis. அப்ப DNA duplicate ஆகவில்லை அப்படின்றதை நம்ம நல்லா மைண்ட்ல வச்சுக்கணும். There is no DNA duplication. So interkinesis only is happening. Yes, Ahmed, yes, Tiruvira and Subhan, very correct answer. There won't be any interface present between prophase 2 as well as between telophase 1. So there will be interkinesis present. Interkinesis is the resting condition. ஒரு சின்ன கேப் அவ்வளவுதான். நம்ம வேற எதுவும் அங்க பண்ண மாட்டோம். We are not synthesizing DNA duplication எதுவும் நடக்காது. இப்ப இங்கே நமக்கு prophase 2, metaphase 2, anaphase 2 and telophase 2 எல்லாமே இருக்கு. It is equatorial division. இல்லையா? So equatorial division அப்படின்னு சொல்றோம். நம்ம ஆல்ரெடி மைட்டாசிஸ்ல படிச்சதுதான் இது equatorial division அப்படின்னு சொல்றது. So prophase 2ல உங்களுக்கு என்னெல்லாம் நடக்கும்? Centriole பாத்தீங்கன்னா it is migrating to the opposite pole. And then spindle fiber attached. And then membrane disappear. Cytoplasmic organelle disappeared. So all these changes are happening in prophase 2. Okay. Metaphase 2, they all aligned in the equator. Of course, it is what we call it as metaphase plate or equator plate. Yes, Yogeshwari, very good evening. Hi. Very good evening, Yogeshwari. We were at the most end of the class, Yogeshwari. Happy to see your name, Yogeshwari. So metaphase plate or equatorial plate, they are all aligned in the equator. Okay, that is metaphase 2. இந்த இடத்துல நான் ஒரு question கேக்குறேன். இப்போ நமக்கு suppose 250 pollen grain நடக்கணும் கிடைக்குது அப்படின்னா எத்தனை meiosis நமக்கு நடந்திருக்கணும்? Shall I ask you 200 otherwise? 400 pollen grain. To get 400 pollen grain, how many meiosis we need? Yes, very good evening, Yogeshwari. Very happy to see your name. Yes, 400 pollen grain form ஆகிறதுக்கு எத்தனை meiosis தேவைப்படும் என்பதுதான் கேள்வி. எஸ் திருவீரா, அகமது, யோகேஸ்வரி, சுபான், புஷ்பகுமார், anybody? Dharni? 400 போலன் கிரெயின் உருவாக்குறதுக்கு எத்தனை மியாசிஸ் நமக்கு தேவைப்படுகிறது? ஆப்ஷன் கொடுக்கவா? 50, 100. Okay. Yes, Ahmed, it is 100. Yes, Tiruvira, very correct answer. Yes, Dharini, very correct answer. Excellent answering. 100. You all know, ஒரு மியாசிஸ்க்கு நமக்கு 4 கிடைக்கும். Yes, Yogeshwari, very correct answer. Okay. ஒரு மியாசிஸ்க்கு 4னா அப்ப 400 செல் இருக்குன்னா நமக்கு 100 மியாசிஸ் நடந்திருக்கு அப்படின்றது கரெக்டான ஆன்சர். வெரி கரெக்ட். எக்ஸலென்ட் அகமத், திருவீரா, தாரணி and Yogeshwari and Tarunika, very correct answer. Now next question, same way, to get 100 ova or egg, we need how many meiosis? இப்ப 100 ஓவா கிடைக்க நமக்கு எவ்வளவு மியாசிஸ் தேவைப்படும்? சாரி 400 வச்சுக்கோங்க. 400 ஓவா கிடைக்க எத்தனை மியாசிஸ் தேவைப்படும்? 100 நினைச்சிட்டீங்களோ? 400 ஓவாக்கு எத்தனை மியாசிஸ்? ஓகே 100னா 100 போட்டுட்டீங்களோ? Okay. 100 na 100, 400 na 400 is the very correct answer. Excellent answering by all of you. 100% correct. Tiruvira, Dharini, Subhan, yes, Ahmed and Subhan, it will be 100 or 400, Subhan. Yes. So, you know, meiosis time la namakku 4 kadikkum, but female gamete la out of 4, 3 will be denatured. Only 1% அப்படின்னும்போது நமக்கு அங்கே வந்து 400 மியாசிஸ் நடந்ததுன்னா 400 கிடைக்கும். 1000 மியாசிஸ் நடந்தா 1000 தான் கிடைக்கும். இப்ப சப்போஸ் நம்ம இப்படி கொஸ்டின் போடுறோம்னு நினைச்சுக்கோங்க. டு கெட் 200 சீடு எவ்வளவு மியாசிஸ் நமக்கு தேவைப்படுது? டு கெட் 200 சீட்ஸ். Yes, 3 cells degenerate every time in female gamete alone, not in male. அது ஞாபகம் வச்சுக்கணும். இப்ப 200 சீட்ஸ்னா yes, Yogeshwari, very correct answer for the previous one. 400 meiosis for 400 ova is the correct answer. இப்ப கேட்ட கேள்வி என்ன அப்படின்னு பாத்தீங்கன்னா நமக்கு 200 சீடுனா நமக்கு என்ன வந்திருக்கணும்? 200 ஸ்பெர்ம் இருக்கணும், 200 ஓவா இருக்கணும். சோ 200 ஸ்பெர்ம் ஃபார்ம் பண்ண எத்தனை மியாசிஸ், 200 ஓவா ஃபார்ம் பண்ண எத்தனை மியாசிஸ் தேவைப்படுது? Yes, Tiruvira, excellent answer. Very correct. Very correct. It is 250. Yes, Tarunika and Subhan, excellent answer. It is 250. So, you know, for getting 200 sperm, you need 50 meiosis needed. 15 to 4, 200. But for ova, you need 200. So, total it will become 250 meiosis we need. Yes, Agamed. Very correct, Yogeshwari. Very correct answer. Excellent. Okay, now it is story time before we complete actually. So, இன்னிக்கான ஸ்டோரி என்னன்னா நிறைய பேர் என்கிட்ட சொல்றாங்க நான் படிக்கிறேன் மறந்து போயிடுது, எவ்வளவு நேரம் தான் படிக்கிறது, நெகட்டிவ் வருது, இதெல்லாம் அவாய்ட் பண்றதுக்கு ஒரு சின்ன குட்டி கதை நான் சொல்றேன். ஆக்சுவலி உங்க எல்லாருக்கும் தெரிஞ்ச கதை தான் இது. இட் இஸ் நாட் தட் நியூ. ஸோ இன்வால்மெண்ட் அப்படின்றத பத்தி தான் இந்த கதை. இன்வால்மெண்ட் என்ன ஆர்வம். ஸோ ஒரு விஷயத்த நம்ம ஆர்வமா பண்ணும்போது நமக்கு நேரம் போறது தெரியாது, தூக்கம் வராது, பசிக்காது இல்லையா? சுபான் கிரிக்கெட் விளையாடுற மாதிரி எடுத்துக்கலாம். சுபானுக்கு கிரிக்கெட்னா அவ்வளோ பிடிக்கும். ஆனா பாருங்க இன்னைக்கு சுபான் வந்து ஃபைனல் மேட்ச் கூட விட்டுட்டு இஸ் லிசனிங் த கிளாஸ். I am so surprised that Subhan is attending the class today. Thank you so much Subhan for your involvement first of all. Okay. So அது வந்து என்ன அப்படின்னு பாத்தீங்கன்னா you know about ஒரு சின்ன ஃபேமிலி. அவங்களுக்கு 2 பையங்க இருக்காங்க. Oh, India won the match. So only Subhan. Okay. So happily attending. Yes. Okay. சோ ஃபைன். இப்ப கதை என்ன சொல்லுதுன்னா, ஒரு அப்பா, அம்மா அவங்களுக்கு 2 பசங்க. ரொம்ப புவர் ஃபேமிலி தான். அவங்களால மிடில் ஸ்கூல கூட தாண்ட முடியல. அவங்களோட படிப்பு வந்து பெருசா காலேஜ் எல்லாம் போகல. அண்ட் அவங்க அப்பா வந்து அவங்க சின்ன பிள்ளைங்களா இருக்கும்போது ஒரு பொம்மை வாங்கிட்டு வந்து தராரு. அது என்னன்னா, நம்ம சின்ன வயசுல விளையாண்டுருப்பீங்க நீங்கலாம். லைக் அத உருட்டி விட்டோம்னா, அது கொஞ்சம் பறந்து போகும் இல்லையா? அந்த மாதிரி ஒரு பொம்மைய வாங்கிட்டு வந்து அவங்க அப்பா அந்த பிள்ளைங்களுக்கு குடுக்குறாரு. அந்த பிள்ளைங்களுக்கு அது ரொம்ப பிடிச்சிருச்சு. இந்த மாரி நாமளும் பறக்கணும் அப்படின்னு அவங்க நினைக்கிறாங்க. ஆனா அவங்க கிட்ட படிக்கிறதுக்கே வசதி கிடையாது. அப்புறம் அவங்க என்ன பண்றாங்க, தே ஆர் ஸ்டார்ட்டிங் ஒன் சைக்கிள் கடை. ஓகேயா? சோ சின்ன ஒரு சைக்கிள் கடை அவங்க ஆரம்பிக்கிறாங்க. அந்த சைக்கிள் கடையில இருந்துட்டே ஒரு 6 டூல வச்சுண்டு அவங்க என்ன பண்றாங்க, தே ஸ்டார்ட்டட் அரேஞ்சிங் அ கிளைடர். அஃப்கோர்ஸ் உங்க எல்லாருக்கும் தெரிஞ்சிருக்கும் நான் யாரோட கதைய சொல்லிட்டு இருக்கேன் அப்படின்னு. அவங்க வந்து ஒரு கிளைடர டிசைன் பண்ணியிருக்காங்க. அந்த கிளைடர் டிசைன் பண்ணும்போது அவங்களுக்கு நெறைய வாட்டி ஃபெயில் ஆகுது. அவங்க தூங்கல, சாப்பிடல, ஏன் கல்யாணம் கூட பண்ணிக்கல. அவங்க முழு நேரமும் இதேதான். அவங்களுக்கு அதுல ரொம்ப இன்ட்ரஸ்டடா இருந்ததுனால, வேற எதுலயுமே கான்சன்ட்ரேட் பண்ணல. ஓகேயா? சோ இதுலயே அவங்க டைம் ஸ்பெண்ட் பண்றாங்க. அஃப்கோர்ஸ் அவங்க நெறைய படிக்கல. இதையே படிச்சு பெரிய ரிசர்ச் பண்ணவர் என்ன பண்ணாரு, அவர் ஒரு கிளைடர் டிசைன் பண்ணாரு. அந்த கிளைடர்ல அவர் பறக்கும்போது அது கீழ விழுந்து அவருமே இறந்துடுறாரு. இந்த நியூஸே உங்களுக்கு கிடைக்குது. பெரிய படிப்பு படிச்சு ஆராய்ச்சி பண்ணி அதுல பண்ணவரே விழுந்து செத்துட்டாரு அப்படின்னும்போது நம்மளால சக்சஸ் ஆக முடியுமா அப்படின்னு அவங்க யோசிக்கவே இல்ல. அவங்க 6 டூல வச்சு பண்ண கிளைடர் தான் நம்மளோட ஏரோப்ளேனுக்கு பெரிய மாடலா இருந்தது. அஃப்கோர்ஸ் உங்களுக்கு நிறைய பேருக்கு தெரிஞ்சிருக்கும். ஐ எம் டாக்கிங் அபவுட் ரைட் பிரதர்ஸ். கரெக்டா கண்டுபிடிச்சிட்டீங்களா? எஸ்ஸா? சோ அந்த ரைட் பிரதர்ஸ் வந்து அவங்க பெருசா டூல் எல்லாம் வச்சில்ல, ஏரோநாட்டிக்கல் இன்ஜினியரிங் படிக்கல. 6 டூல வச்சு ஒரு கிளைடர் ரெடி பண்ணாங்க. அஃப்கோர்ஸ் அது வந்து டிசம்பர் 17 19 some 1893 ஐ திங்க் so. நாட் வெரி ஷுர் அபவுட் இயர்ஸ். எஸ், ரைட் பிரதர்ஸ், ஆர்வில் அண்ட் வில்மர் பிரதர்ஸ். வில்மர் ரைட் அண்ட் ஆர்வில் ரைட் அவங்க 2 பேருமே அப்படி டிசைன் பண்ண அந்த கிளைடர் தான் பின்னாடி வந்து அஃப்கோர்ஸ் ஏரோப்ளேன் மாடிஃபை பண்ணி கொண்டு வந்தது. நம்மளால மனுஷங்களையும் தூக்கிட்டு பறக்க முடியும் அப்படின்றத ட்ரீம் பண்ணவங்க அவங்க 2 பேர் தான். சோ இந்த கதை எதுக்குன்னா இன்வால்வ்மெண்ட். சோ நமக்கு அது ரொம்ப பிடிச்சிருச்சு அப்படின்னா நம்ம அதை கண்டிப்பா செய்வோம். அந்த பிடிச்சதை எப்படி நம்ம ஆர்வமா ஆக்கிடுறதுன்னா, நீங்க படிக்கும்போது எதெல்லாம் கஷ்டமா இருக்கோ அதெல்லாம் நீங்க வந்து ஒரு ஃபிளாஷ் கார்டு மாதிரி கொண்டு வரலாம், இல்ல மைண்ட் மேப்ஸ் கொண்டு வரலாம், சின்ன டயக்ராம் மாதிரி கொண்டு வரலாம். இதெல்லாம் கொண்டு வந்தீங்கன்னா இட் வில் பி ஈஸி ஃபார் யூ. ஆர் டேப்லர் காலமாவோ போட்டு வச்சுட்டீங்கன்னா இட் வில் பி ஈஸி ஃபார் யூ டு ரிவைஸ் இன் அ ஷார்ட்டர் பீரியட். ஓகேயா? So I hope this will help you to boost your score, also help you to improve your revision. Yes sir? So இப்ப நம்ம கதையை கண்டினியூ பண்ணி முடிச்சிட்டோம். இப்ப நம்ம ஸ்டோரி வருவோம். பேக் டு த metaphase. I hope you all understood. இப்ப anaphase. So anaphaseல உங்க எல்லாருக்கும் தெரியும், இப்போ சென்ட்ரோமியர் ஸ்பிளிட் ஆறது. சோ சென்ட்ரோமியர் ஸ்பிளிட் ஆச்சுன்னா என்ன அர்த்தம்? குரோமோசோம் டபுள்ஸ். ஓகேயா? அப்போ இப்போ நீங்க எனக்கு சொல்லுங்க, ஆல்ரெடி we know we had 23 chromosome, 46 chromatids. So 23 chromosome இருந்தது, 46 chromatid இருந்தது in anaphase 1ல. This is in anaphase 1. இப்போ anaphase 2ல number of chromosome and chromatid, can anybody share the answer to me? Number of chromosome and chromatids in anaphase 2. Who will answer me? Subhan said 46. Subhan, already chromosome 23 only, Subhan. யோசிங்க குரோமோசோம் டபுள் ஆகும், அது கரெக்ட். குரோமாட்டிட் இல்ல, Subhan is correct only. நான் சும்மா twist பண்ணேன். Yes, it is correct answer by Subhan and Tiruvira. Excellent. Excellent answer. Okay. So 46 chromosome தான். ஏன்னா chromosome double ஆயிடுச்சு இல்லையா? So we have 46 chromosome. Chromatid double ஆகாது. ஏன்னா நமக்கு interphase நடக்கல. அதனால 46 chromatid தான் இருந்தது. So we have 46 chromosome. Okay. Chromosome and 46 chromatida இருக்கும். Yes, Subhan. Very correct. So, இப்போ இது வந்து என்ன ஆகும்? Late anaphaseல உங்களுக்கு தெரியும் ஒரு furrow ஆகும். அது split ஆகும். அப்ப மேல உங்களுக்கு 23 chromosome and 23 chromatid. கீழ 23 chromosome and 23 chromatids ஆகும். அதுதான் வந்து telophase 2. So, you have 23 chromosome and 23 chromatids. Okay. So telophase 2, of course, they are haploid in number and you are getting 4 number of cells in telophase. So the overall step, உங்களுக்கு தெரியும் prophase 1ல நமக்கு நிறைய ஸ்டேஜ் இருந்தது. லெப்டோட்டின், zygotene, pachytene, diplotene, diakinesisல crossing over எல்லாம் முடிஞ்சு they aligned in equator. இங்க நமக்கு சென்ட்ரோமியர் ஸ்பிளிட் ஆகல, அதனால ரிடக்ஷன் பாதி பாதியா பிரிஞ்சிடுது. So each cell will receive 23 chromosome and 46 chromatids. And the 23 chromosome and 46 chromatid when it undergo prophase 2 and metaphase 2, anaphase 2ல splitting of centromere நடக்கும். அதனால chromosome double ஆயிடும். 46 chromosome and 46 chromatid இருக்கும் each cellக்குள்ள. அது தனித்தனியா பிரியும் போது 23 chromosome and 23 chromatida மாறிடும் என்பது சரியான பதில். Very correct answer. Okay. So what are the significance of meiosis as we discussed already? Crossing over நடக்குது இங்க. Shuffling. இந்த crossing over நடக்கிறதுனால variation நமக்கு தெரியறது. So variation இருக்கிறதுனால of course it is a raw material for evolution. Okay. And then it maintain constant chromosome number. It maintain constant chromosome number. These are all the advantage. இப்போ இத base பண்ணி can you all answer me what is the chromosome number and chromatid? Chromosome number, chromatid value numbers in primary spermatocyte, secondary spermatocyte, and sperm. So இது மூணுத்துக்கும் எனக்கு பதில் சொல்வீங்களா? Primary spermatocyte ஓட chromosome number, chromatid number, secondary spermatocyte ஓட chromosome number and chromatid number, sperm ஓட chromosome number and chromatid number. So what would be your answer? For all the three. Yes, I am waiting to receive your answer, children. What is the chromosome number and chromatid number of primary spermatocyte, secondary spermatocyte and sperm? Any guess? Anybody is working? Chromosome 46, 23, 23. Very correct, Subhan. Excellent. Chromosome number is very correct answer. 100% correct answer. Chromatid, can you say? So yes, primary spermatocyte is 2n, so 46. Secondary spermatocyte haploid 23. And sperm also 23 haploid. So chromatid. Yes, Yogeshwari, chromosome is very correct. You are thinking, Subhan? Okay, primary spermatocyte is 2n 46 chromatid means you will have 46 chromatid only. Secondary spermatocyte, of course, it will undergo interphase, so it will become 92 in the middle. So you will get same 46 here in each of the cell. Then after sperm, it will become meiosis 2, it will become 23 only. Yes, sir? So 46 2n undergo interphase and forms 92 during S phase. When it undergo meiosis 1, at the end haploid cell, each one receives 23 chromosome, 46 chromatid, and sperm after meiosis 2, 23 chromosome, 23 chromatid. I hope you all understood. This is another possible question from meiosis. Little critical question. You may get like this also. So any other doubt? I hope the today class you all enjoyed. So I also gave you some motivation tip, and you can prepare a flashcard or tabular column or highlighting or a tabular column or flashcards anyway. You can, or mind maps, you can prepare and you can make it easier and do well. Okay. Thank you all for participating, every one of you. Any doubt, please stay back and ask me. If not, we can finish the class. Thank you all for everyone. Yes, thank you, Subhan. Thank you, Tiruvira. Thank you, Keshwari. Thank you, Dharni. Thank you, Subhan. Thank you, Tiruvira. Thank you, Dharni. Thank you, Ahmed. What is nicking? See, exchange happens now between A chromosome and B chromosome. Thank you, Yogeshwari. A and B. The part of A chromosome will move to the B and part will come B from A. Exchange happens now. I showed you in the picture. If you see, the blue comes to red, red comes to blue. This is what we call it as nicking, the half exchange. Yes, happy Vinayaka Chaturthi to everyone. Yogeshwari, thank you for wishes. Thank you so much. Tiruvira, you understood what is nicking now? That a part of the segment is moving from here to here. So this blue has gone to the red one and red has gone to the blue one. I hope you can see in the PPT now. Yeah, thank you very much. Yes.